seeds that become very, very abundant in ecosystems that establish so well in a different place than where they're originally from, we have a few hypotheses in the field. So I'm going to go through these relatively briefly, but I'll walk you through. So this is the outline of the rest of my talk. I'll walk you through what I see as the three major hypotheses we have for what might be special about these species and different than the species in the communities in which they've arrived. And then I'll dig into some of my research, which will touch on some of these major concepts as well. So the first one that I'll talk about is the idea that invaders might come in with a novel niche. So what do we mean by a novel niche? The hypothesis here is that some invasive plants are super successful because they do something that's really different than other species, and in particular, this gives them access to resources that other species don't have. So it might be some kind of different type of resource that they utilize that they can use that other species can't. It might be more, and in plants, they mostly need the same sort of basic requirements of light and water and nutrients. And often what we're talking about is being able to use those in a different space or time than other species use them. So they do something in a different way. And this gives them their own unique edge in the new habitat. An interesting example of that, legume species are one of the characteristics of this group that makes them special and really interesting is that it's a major group that associates with bacteria and these little nodules in their roots, and they can make their own fertilizer. They can take nitrogen from the air, and in this little partnership with bacteria and the roots, they can make their own fertilizer. And this is a picture of mesquite out here in the Sonoran. Lots of our Sonoran species, our trees especially, are in the legume family, and they do this. They make their own fertilizer. This is a very useful set of adaptations. It gives these species a certain niche where they tend to come into very resource-poor ecosystems that don't have a lot of fertilizer, natural fertilizer, don't have a very rich soil to start with, and these species do really well in these kinds of environments. And we see this all over the place. Outside of places like deserts, in other places, we tend to see these kinds of species come in after a disturbance, or maybe you said there's been a flood or something, and there's open space and bare ground, and these are some of the first colonists that come in because they have this ability to make their own fertilizer. Okay, so that's just something that this is a really in plants. This is one of our really obvious examples of species that have a particular niche that they come into. Well, to mention Hawaii again, Hawaii actually turns out that they do not have species that do this nitrogen fixing in these young soil environments that are very resource poor. They just don't have species that fill that kind of role of colonizing these disturbed spaces. And so what happens, there are species that have been introduced that are nitrogen fixers that do this kind of making their own fertilizer, and they have come in and invaded these environments. And like Buffelgrass with its promotion of wildfire, these also really altered the ecosystem because they produce all this nitrogen, and then they make the environment much easier for other species to come in behind them. That is not the normal progression of stuff that you find on the highlands. So this is an example we think of the kind of thing we mean by a species coming in with a novel niche. And I think in plants, this is happening all the time in much more subtle ways that are a little bit harder, a little bit less obvious than species that can fix nitrogen or can't. Ways that maybe are a little bit harder to see, but again, somehow species are doing things a little different in space or time or resource use. In contrast, we also have a concept called novel weapons, that species come in with unique weapons. What am I talking about? Plants can have weapons too. All right. So the hypothesis here is that invasive plants can fight off competitors or enemies, and the ones that become invasive might have special ways to do this that other species can't. Okay, they are coming in with a new mechanism. You can imagine, for instance, a species that's unpalatable to insects that might chew on it, or something that has some kind of new compound in it. None of the insects in the resident environment can eat it. I'll give you another example. Some of you may know, in much of the rest of the country, not right here, garlic mustard is a species that's incredibly successful. So this is a Eurasian species that's been brought over. And what garlic mustard does that's interesting is that it produces a toxin, at least one. It produces some toxins in its roots that go into the soil. So it's a compound called sinigrin that is toxic to other plants. and that seems to be one of its components of success. So this is a forest understory species. And one of the ways that we can tell that this kind of weapon that it has is important in these systems is seeing the evidence of actually plants adapting to these different conditions over very short time scales. So what I'm showing you here is some really interesting data from a paper by a colleague of mine, Richard Lankow. And this is showing that, okay, so in different populations, if you look at how much synegrin these guys are making, so how much of their weapon they're making, they make more of it the more other kinds of plants are around. And an opposite way of looking at that is, in fact, we can see after these guys compete and they outcompete other species, and you end up with populations that have almost no other species in them, these populations have actually been shown to evolve lower levels of production of this compound because it's not useful anymore and it's costly for them to make it. So it's really fascinating. In the span of about 150 years that it's been here, we see evidence that these populations are evolving different levels of chemical warfare depending on the context. In response, this is tolerance of native plants to competition with these guys. If you look at the synegrin that's in the soil at the source population and you look at the level of tolerance, they're actually, these are older plant populations where there's a lot of synegrin that's built up. The native plants that are there are showing evidence of evolution of more tolerance of this compound in the soil. So these guys are, it's not an arms race so much as they're duking it out. They're probably gonna cycle around for a little bit. This is dynamic, but it seems to be a very important part of what's happening in these populations and an important part of why this is successful. Finally, we have a hypothesis that's probably one of the most prominent hypotheses in the field, which is called enemy escape. And this is an intuitive idea that's been a bit sticky. So the idea is, the hypothesis is that invasive plants have left behind in their former range enemies of some kind. It could be diseases, it could be herbivores, could be all kinds of things, or the combination. They used to reduce their populations. They've left those behind. And in the new environment, they don't have new effective enemies yet. So there are not diseases that are evolved to attack them, or the herbivores in the new range don't recognize them as food yet. So something has happened here that they have left enemies behind. They have not gained substantial new enemies. It's an intuitive idea. We see, the upshot of this is, okay, first of all, it's just good for the invaders if they're experiencing less attack by these things. So they lose less fitness to that. The other thing that can happen is that then it is possible that they can either develop or even populations may evolve over time to invest less in the defenses that plants can make against these things. If they're not getting attacked, then they don't have to invest. A little bit like that garlic mustard not investing in that chemical that it uses for competition. Here we're talking about defenses against enemies. So we think this is all possible. It's very hard to show, but I'll comment on that. So this is a bit of a holy grail. Oh, I'm sorry, this got a little messed up. This says native range and then invaded range. This is a nice cartoon a colleague of mine made. This is, to illustrate this idea. So the idea is that in the native range, you have a well-defended plant that is not quite as good at growing because it's investing a lot in defense, maybe less in growth and reproduction, but it's doing so because it's getting attacked by the dapper yet problematic herbivore. And then you move it to a new place. I'm free to do what I want any old time. So now this guy isn't here. Nobody recognizes this. No need for defense. Resources are available for growth and reproduction, either immediately or the plant actually can evolve to this new situation, is the idea. The interesting thing about this is that it doesn't require special traits of any of these plants. This could be, if this is true, that this is one thing that may happen to plants, it could happen to lots of different plants, to all the different things that are coming in and invading. So it's appealing as a very broadly applicable hypothesis. They don't need any special traits of the plant or the community that it's invading. Also, this is the underlying hypothesis really that's at the heart of the idea that biocontrol should work, that we should be able to find things in the native range, bring them over, and suddenly we'll restore this balance and these guys will be kept in check. That's the idea. Of problem with this very appealing hypothesis is that it's very, very hard to actually demonstrate that it's happening. There are many, many things that can nail a plant, diseases that might affect it, herbivores that might attack it, and they don't happen every year. If you want to go outside and see what the important ones are, that's a very difficult task to do, to see things microscopic and macroscopic that may be ephemeral over time. And then you have to see that those interactions have changed. You've got to do this probably on 2 different continents to see that those things are different. And then you have to be able to say that that difference is enough to actually make a difference to abundance and distribution, if that's actually going to explain the advantage that the invader has. So it's been a bit of a challenge. It's a long-standing, very appealing hypothesis is a challenge to show that it's actually true. Indirect evidence suggests that it's likely that it is true, at least in some cases, because, for instance, we have some really good cases of effective biocontrols. One nice example of that is Klamath Weed, Hypericum perforatum. This, in the western U.S., was a major problem early to mid-1900s. So it was a major pest of grasslands, and in fact, one of the things that people like to quote about this invasion is that there came a point, I think it was in about 1947, when landowners in the West that had this plant on their land could not get a loan using the land as collateral because it was considered so devalued. So incidentally, cattle don't like to eat this, and it has some toxicity. So this was a very bad infestation. Right around 1947, the biocontrols, little flea beetles were introduced. Chrysalina species, and particularly Quadrigemina, was very well established, and within 10 years, this was 90% knocked back. It was an amazingly successful biocontrol. One of the interesting sort of fun facts about, I think, why it works so well is that the beetles really nail the plants in the sunshine. If they're in a little bit of shade, the beetles don't do as well. And so it kind of kept a reservoir of a few plants around, kept the beetles going, and you kind of hit that perfect sweet spot where the biocontrol stays in the system, the beetles can persist, the plants can persist, but everybody's at a low level. This is the dream of biocontrol. There's lots of failures and lots of cases where things don't work very well, but this is a poster child for it working. But it does suggest that one of the advantages this plant had when it came and exploded in abundance was escaping these guys that were adapted to attack it, and that they do a good job of attacking it. Okay, so those are some of our major hypotheses for major categories of why invaders might be different than natives and have particular advantages: novel niches, weapons, and enemy escape. So I'll tell you a bit now about research in my own lab where we're trying to tackle some of these questions. All right, so we work on a variety of things, and our goal is actually to bring together ecology, studying how organisms are interacting with their environment, but also to use the tools of genetics and look at evolution of genetically based traits to try to understand how these different environments affect the distribution of plants. A bit like that example with the garlic mustard that I told you about, where we can watch the evolution of populations that tells us a lot about important things that are happening in the system. So we do that in my lab. One of our focal species, we work on a few things, but we spend a lot of time these days working on this particular species. This is yellow star thistle, or Centaurea solsticialis. It's a grassland thistle species. It's considered one of the 10 worst weeds of the West. So it's out in the western grasslands. Cattle don't like to eat it, and it makes horses sick. So, and it's a Eurasian species, so it's been introduced. The distribution is broadly Eurasian, has a broad native range, and then it has been introduced into the Americas, where we have problematic introductions. The history is that this plant was historically a weed of alfalfa fields. So we think in the old world, when the Spanish colonized the New World, we know that they brought alfalfa cropping with them. So they established alfalfa farming. And so how that would have worked was to let your field go to seed to get your alfalfa seed to bring it elsewhere, and you would have gotten all of the wildflowers that were in there that went to seed around the same time would be in your seed lot. And so we think that is how it traveled first to South America and then to North America with the Spanish. And Then in the 1900s, with irrigation, the West was really opened up to a lot more agriculture, and there was a lot of interest in expanding alfalfa cropping. There was not enough local seed. We know that there were additional seed lots that were imported from across Eurasia, and in those days, they actually kept records of the weed seeds that were in those seed lots, because no one has ever liked getting weeds in their crop seed, and so they actually tallied that. So we know that yellow star thistle was in those seed lots, as well as a who's who of all of the other things from Europe that are in our grasslands out west. They were all in those seed lots, and they probably had been since the beginning. There's a map, I think, on the back, but in California, where there's some good records, so the first North American record of this plant was in the late 1800s. There's some records of it on the edges of agricultural fields in California. In the late 1900s, it moved into grasslands and just exploded in numbers. So this is an area infested in millions of acres. And here's the distribution. This is not current. This is from 2003, but this is the extent of the distribution in California, which has a major battle with this weed. Incidentally, there are 6 biocontrol agents introduced so far, and they haven't controlled it. So, no Klamath Weed story there. And this is what the problem really is. So that other one was a pretty picture with some cattle in it, but these pictures, everything yellow in these pictures, this is Argentina, this is California, Idaho also has a problem, as well as Eastern Washington and Oregon, and these are bad infestations. Just amazing. Interestingly, these kinds of situations are not what you see in the native range. If you go to Europe and you look for this plant, you find it as an incidental, and people have measured this, so this is density of plants in California and Argentina invasions. It's more than an order of magnitude less in Europe where people have measured it. So this is a really different ecology. Something really different is going on with these plants in the New World. Okay, so maybe a novel niche. What are these guys doing? I don't know. Let's find out. So one of the things that's really interesting to us is that there had been a report, and we grew plants, and we could see this very clearly. If you grow plants, these are plants that I have grown in an experimental garden in a greenhouse, and I planted them at the same time. They have the same resources. This is one from the native range, and this is one from Central California. This guy has much more biomass, more root biomass, more shoot biomass, longer, wider leaves. This vertical growth is bolting. It's starting to flower much earlier than this one, and in the course of the experiment, this will make a lot more flowers. So this guy looks like he's just on steroids. He's doing everything better with the same resources, growing at the same time. So how on earth is this happening, and how do you get from here to there, and why? If we can answer that question, we might learn something about why this is so successful. So we have collected these plants from populations from all over the world and grown them in gardens to study these differences, and this is just to show you a little bit more of that data. Invaders are definitely larger. This is just showing you temperature seasonality of the environments that these populations came from. So this is a little bit about how the habitat is different. We would expect populations in different places might have little differences in their traits, we call local adaptation. And this is a size index on a log scale. These are the invaders from California. These are the ones from our collections from across Europe. So in the same kind of environment, these guys are much, even controlling for that, these guys are much bigger. And at the time we did this, we didn't know what the source was for these in terms of who their progenitor populations were. We now know that, and they're the ones right down here. So it's actually where the difference is the biggest. They're from similar environments, but they're hugely different. This leads to, this difference in size leads to earlier flowering. This is days to flower, and even controlling for latitude from where the populations came from. In Europe, there's a nice latitudinal change in when populations flower. They're adapted to their different latitudes. In California, everybody just flowers earlier, and this leads to more flowers. The other thing it leads to, these bigger plants that are doing more growing and more reproducing, is that they need more water. So we did some drought tests, and we looked at how long they can function before they wilt, and the invaders wilt much sooner than the native plants, and this is directly related to size. So they're bigger, they need more water. This is really interesting because in the California grasslands, there's a lot of good ecological work, and when these guys are out in the summer, water is really the limiting resource, and the plants that are out at that time compete very strongly for water. So these guys have high water needs. but they have evolved a phenotype of traits that make them really demanding of water. And that's what I was going to say there. So in this environment, water is the key resource. So what we hypothesized in a recent paper was that what we were really seeing here was a species that in this system, there's reason to believe that there are not good competitors largely for water anymore. Because of all kinds of ecosystem change in the California grasslands, there are not other wildflowers that compete for that late season water. And that this opened up a niche. So it's not exactly a novel niche, it's kind of a vacated niche. There are no native species using that water anymore. So these guys came in and it's just like bonanza. And there's no competitors for water. So that was that's the hypothesis that we put forward, that these had evolved this very aggressive, these big size, big traits, that they could do this because they weren't competing with anybody for water. And I still think that's true, but there's something that kind of bothered me about this. Okay, so it suggests escape from resource competition, from competition for water, and that these guys can grow big and hungry. But these are the hypotheses that we're testing based on looking at the system and kind of our best ideas about what's going on and just thinking through it logically, but it doesn't mean that we're asking the right questions. There are so many different things we could study about these environments. How do we know that we're looking at the most important things that have changed and that have given rise to a situation where this species can do so well? And so that kind of bothered me. So maybe we have a novel niche situation. Let me turn to a different topic. Maybe it's not, maybe it's something else. So the approach that we took is, this is where the genetics comes in. It's possible actually to study the genome of a species and look for evolutionary changes between populations in the content of the genome. And you can actually use that to make some inferences about what kinds of ecological traits might have changed. And I'll walk you through how we've tried to do that. And this is an approach that was coined reverse ecology, which is because you don't start with the ecology, you get there at the end. You start with the genomics. And it's a very interesting idea, and one that was very appealing to me because I was concerned about whether we were asking the right questions. So how we do this is we look for the evidence that there's adaptation. And there's many ways to do this. There's several ways. This is the way that I'm going to talk about today. The idea is that we look at genetic variation in the source region, so the native range, in Europe in our case. Imagine that each one of these things is a genome, and the little dots are different mutations that different individuals happen to have. And I've colored some of these bright blue so they stood out. Imagine that there's some mutation hanging around in the native range that's maybe not very common, and it's not very useful in this range, but it's going to be very important in the introduced populations. Then what we can do is we can go to the introduced populations and look for situations where we see a particular variant is now present in virtually all of the plants that are there. And recombination, that mixing up of our genetic material that happens like between our parents, that will happen at other sites away from here. So they'll have lots of different variants from the native range at other sites, but maybe there will be particular spots where we'll see only a few variants. We'll see a signature of really low diversity there. That's what we look for. So we don't see much variation. It might mean that we're close to some kind of site that's been favored, that particular mutation is favored. Basically, at this location in the genome, everybody's a descendant from these original guys that carried it. To do that, we had to figure out exactly which population in the native range to compare to. So if we're looking for some kind of variant that started out less common here and is now characterizing invading populations over here, we need to actually know where we should sample from in the native range, because there's lots of different variants across this huge geographic space. So we did that. If you're interested, if you're not, just look at a saguaro while I go over that. But what we do, this is the genome. What we do to get markers to figure out, to get information from the genome to figure out who's related to who, we chop up the genome with restriction enzymes, so they recognize a particular sequence of DNA and they will cut the DNA at that position. That gives us a little position we know the sequence of, and we can actually use that information to attach some little adapters, some little stretches of sequence that we know exactly what the sequence is, so we make that region that we know a little bit longer. And then we select out some fragments, and we can use, so when we sequence DNA, you have to start with a little primer that attaches to some part you know. We can start from the parts we know, and we can sequence into the parts that we don't know. And we can do this for many, many hundreds of individuals. We've got about 1,000 individuals genotyped this way, and we get thousands of fragments across the genome that we get little bits of information about what everybody's sequence is. You know, in 2 years, I'll just give this talk and say we have the complete genomes for everybody, but at this point, we can't do that. That's still too hard and too expensive to do for 1,000 star thistle individuals, about which no one cares as much as humans. But even still, it's expensive. So when we do that, what we can do, these little colors show that we can see different genetic variants in different regions of the distribution of the plant. That tells us something about how different genotypes, different different mutations, different sort of groups of related individuals are spread across space. So we can group them into different regions, and we can start to ask what the family tree is that explains the history of these different regions of related individuals in the native range, which is all these little trees here. These are family trees for the native range, and then we can stick on all these little codes are our invasions, and we can figure out where introduced populations came from that history. And the short answer is that the Spanish introduced yellow star thistle to South America and then to North America. And the genetics say what the history said, which is actually really great. So it is Western European genotypes that have been introduced into South America and then into North America. And these now all have their own genetic compositions, but we did this careful model testing with all of this genetic information, and we were able to sort that out, which people had not been able to do before, because it was a little bit, turned out, for a variety of reasons, it was complicated in this system. And we also know how the parts of Europe were related to each other, which is really interesting. So now we know that indeed we can be sure that it is the Western European plants that gave rise to our invasions. So if we want to ask whether there's some variation here that seems to have been selected here, we know what to compare to now. So now we can do this. Now we know this is Western Europe that we have to ask about, and these are our invasions. So we did that. To get our genetic variation, we used a little bit of a different technique. Here we used express genes. So we're actually taking RNA out. So DNA, when it makes proteins, is transcribed into RNA, and then it's turned into proteins. And so when a plant is growing, and we took actively growing seedlings, the shoots, the leaves, and the roots, it's expressing a lot of its genomes, using a lot of those genes. So we extracted those genes, and then we could get the sequence of those. And this is a way that we can get information about, particularly about the parts of the genome that make proteins that matter to this plant. 5 minutes? Okay, I can wrap up. I have like 5 more bullet points, but that's okay. This is the key part of the story. So when we do that, we can plot out, each dot here, which is not showing up very well, is diversity in the native range for a particular gene, and then we can get diversity in the invaded range for a particular gene. And they're roughly sort of correlated, which you would expect. These guys are from here, and a lot of that diversity was introduced incidentally. So they're correlated. One generally predicts another. But there's a set of these out on the edge here that are particularly low diversity in California for what we'd expect from the native range. And these are our candidates that might be involved in adaptation. And then we can see, especially by alignment to other species like Arabidopsis that have been studied really well, what those genes do. But I'll get you to the punchline. When we look at our list of genes, we see a number of genes that are involved in defensive functions in the plant. So we had been studying competition and resource use, and then we go to the genome and we say, what's changing in the genome? It's defense genes. And that was not something we had been studying. So it was a real eye-opener and really interesting. And so we and suggested that something different was important to the invader. Now this could be a totally separate thing from the growth that we were studying, but it also could be directly related. So plants have a very strong trade-off between investment in defense and investment in growth. If the evolution that we're seeing is actually plants evolving to shut down their defenses, they would automatically become bigger plants. So it may be that this thing that we were studying, being a bigger plant, is actually related to defensive functions. Or it might be that they're totally separate things, and we're trying to test that right now. And the cool thing about this is it could, because we started asking the genome what's been important to natural selection, what's actually selected on these plants, this might actually be the best evidence we ever have that these interactions with enemies are really important. So I'll still try to go out into the field and catalog all the things that are on this plant, but even without that information, I can already tell you that it looks like defense interactions are important to this plant, and we can study that. In traits and trade-offs, I was going to tell you a little bit, we're starting to study immune function. It does look like it's lower in our invading plants, which is what we would predict if they're losing defense function and devoting that resource to size. And in the environment, all these wonderful people doing great work. In the about the environment, we're studying, we have indications that the defensive functions might be related actually to pathogens, and we're starting to study the microbial environment of these plants. It's very different in the two ranges, and in fact, in the invaded range, we find a lower diversity of bacteria on the plants, which we think could be an advantageous situation for the plants. So that's a hypothesis we're trying to test right now. And with that, I'll go to some. So we're trying to make connections between environment, evolution of different genes, traits that they affect, and the abundance that we see in the field. We think there are connections there. And the idea is to understand in general, what is it about these plants, what advantages do they have when they come into a new system? Can we use these kind of analyses to get under the mechanisms of what's going on? So with that, I'll be happy to wrap up and take questions, and I'll just quickly acknowledge lots of people in my lab and wonderful collaborators contributed to this, as well as funding from National Science Foundation, the USDA, and U of A. Thank you.
Awesome. Just before we begin the question and answer, I just want to draw everyone's attention. Last month I didn't have as many of the brochures for the Science Cafe for this season. I have many more on the back table. There are 3 talks just next week and 5 talks in December, so feel free to grab one of those. It's my ultimate hope that I'll have next season's brochure in December, but it's early in December, we're the earliest, so I will probably forward you to our website where there'll be an online version, so you can get those. But I'll have the dates no matter what, so you can mark those. So as we mentioned, please fill out an evaluation. I'd love to give Dr. Dlugosch some feedback, and then also hear from you if you have topics you'd like us to bring to the Tucson Botanical Garden Science Cafe. And so I'll turn it back to you.
Yes. Repeat question. Oh boy, yes.
Do you think what's changed is the gene regulation for these 20 guys, as opposed to the genes themselves changing?
So that's a great question. So the question is, what about gene regulation? Is it the genes themselves that have changed, or the regulation of those genes? So the way in which we did this scan for evidence that there's adaptation in those genes was actually to look at the sequences. So it would suggest that there's evolution in the sequence of that gene itself. But we are simultaneously also starting to work on gene expression, so how much of a gene is being expressed. When we find differences in those, that will be all about how much is being expressed, and we won't know the source of the genetic changes that control that expression. And then we'll try to put those things together. But yes, that... Yeah.
Lots of luck with that.
Well, we have hope. Yeah. Yes.
So what's your feeling? Would it be easier to turn off the defense mechanisms so that other plants could compete, or so that animals would find those plants tasty?
Yeah, so in terms of like a management strategy, so what's my sense of, would it be easier to turn off those defenses? I think, you know, what the most immediate thing that we're interested in doing, because it turned out that the particular genes that came up as being important suggested it might be microbial interactions. One of the things that we're turning our attention to is we've applied for funding to sample microbial environments really broadly in California to see if we can pick up variation within the invaded range itself in environments that are more or less favorable for this plant. Because the idea is that it's the escape that has allowed it to grow into a big plant. So if we can find an unfavorable microbial community to spray on the plants, for instance, they would not be well defended against that. That's the prediction. And we would not want to do that with a microbial community from Europe. I'm not interested in doing that. But there's a lot of variation in microbial communities. It might be that there are communities within the invaded range itself that we could leverage. So there are sites, for instance, where the plant is not doing well, and that's the reason why, and we just didn't know it. We haven't really looked for that yet. So that's what we want to do next, is go out and try to figure out whether we could leverage something like that. I don't think I could engineer the plant, but I could probably leverage the fact that it's given up some of its defenses.
Has that been tested at all with Buffelgrass?
Not that I know of. And so right now, there's lots of work. Has it been tested with Buffelgrass was the question. And I don't know of any programs to do that with Buffelgrass myself. It's possible it's out there. There's a sort of a hot thing going on right now that we recognize that microbiomes can be really important to the function of everything, including plants, including people, to try to use microbial cocktails to control species. And so there are some things coming to market almost with no testing at all. So, it's kind of the wild west out in the west in this area right now. So I have not seen data from anybody that says that something will work like that, but they are showing up. So so you could see something sprayed on Buffelgrass. I would love to see the paper where somebody showed that was what was going on with that interaction, and I think it could work. I'm not saying it couldn't. It's just we're starting to do it without even really knowing. People are saying, okay, here's a cocktail species we know can attack plants in this area. Maybe that will work. And it's kind of like some of the other biocontrol efforts where it's just like, let's put something out and see if it will solve the problem, but it doesn't always. Yeah.
Okay, of the three hypotheses you've named here, which do you think applies most to Buffelgrass and the other weeds?
Of the three hypotheses, which applies to Buffelgrass?
And the other weeds,
So I don't really know the answer to that, but Buffelgrass is an interesting one. It is it's a polyploid. So if you saw the last month's talk about how polyploids rule the world, so maybe that's part of its success. It's also been bred as a forage species. It's had help from humans to be as successful as it's been. That makes me think that while it's possible that it's got a situation like it's escaped enemies, I think it's much more likely that it has some mechanisms of a novel niche, novel way of using resources that allow it to be very successful in arid environments and really be very efficient at converting limited resources into a lot of biomass.
I was wondering if maybe it had could make its own nitrogen, because it grows in places where the soil just looks dead.
Yeah, to my knowledge, I don't think that it can. That ability is very limited to certain plant groups that have the adaptations to do that. So I don't think that it's doing that, although there can be a lot of different microbial associations of species that allow them to access more resources in different ways. Because it's been bred as a agricultural species, and it's not sort of out there, it's not, to have those kind of really favorable microbial associations, you've got to have your microbe moving with you. So it seems like that might be less likely in something that's been widely distributed as a forage species. I think it's more likely that it has to do with the way it uses resources, and it may be kind of subtle things that are not so easy to just point to. I mean, all plants need water and nutrients and such, but it's doing something in a way that's giving it an advantage. And I think that's the ticket. Any other questions?
So in California, all the grasses that are growing there in the summer are invasive also, or most of them.
Yeah, so California is a story of a dramatic wholesale ecosystem change, and that was really part of our original hypothesis about what had gone on. Essentially, there was between overgrazing and drought and massive seeding in of European annual forage species. There was a tremendous conversion in the mid-1900s to European annual grasses in the system. Native species were really knocked back, and they were rare before star thistle took off. And there are native species that can actually compete with star thistle. They're so rare in the system now, they're really not a factor. And probably there's some data now that it's the thatch from the European annuals, so they grow big and they fall over, and the natives are not used to coming up through that. The natives are used to perennial bunch grasses that have a lot more space around them. So that's probably the key to why the natives have not been able to come back. And then something like star thistle, which can come up through thatch, is just free to go wild. Exactly. And it's a very altered system, no question. Well, thank you very much.
Thank you, and I just want to remind everyone, you can get your hand stamped for the garden, the evaluations, and I expect that Ashley will provide some warmers from next month's talk. So don't be dissuaded from coming back, by all means. So thank you and have a great evening. Yeah.